Whether you're a bioprocess engineer hunting for the perfect scaled-down model, a biotech founder architecting the leap to commercialization, or a scientific analyst who craves raw technical truth, you've reached the technical edge. In this space, we live by a single mantra: the process is the product. Welcome to the Biomanufacturing and Fermentation Technology podcast with Prasad Ernala. Hit subscribe and ring the bell to join a global community and stay at the vanguard of biotech translation. Let's build the bioindustrial future together.
Today's part 3 of 4 parts of QbD with the theme of discussion is Making Models Survive Reality. This part outlines how to bridge the gap between theoretical bioprocess models and the practical realities of large-scale manufacturing. It emphasizes that high-quality data and experimental discipline are the foundation of reliable models, particularly when transitioning from small shake flasks to complex bioreactors. The author explains how Process Analytical Technology, PAT, and soft sensors provide the real-time visibility necessary to maintain process control and ensure product quality. Furthermore, the expert advocates for robustness testing to identify stable operating plateaus rather than fragile performance peaks. By embedding scale-up physics and mixing dynamics into early development, engineers can create processes that remain resilient against physical gradients and oxygen limitations. Ultimately, the text argues that integrating mechanistic understanding with rigorous execution ensures that optimized laboratory conditions translate successfully to commercial production.
Welcome to the debate. Okay, picture this. You've spent, 18 months in the lab, maybe more. You've designed what looks like a perfect bioprocess. Your small-scale bioreactors are humming, yields are optimized, and your statistical model, it tells you you've found a robust design space. You're feeling pretty good.
And then you take that process to manufacturing, you dump that delicate biology into a 20,000-liter stainless steel tank. And, well, suddenly your beautiful model isn't worth the hard drive it's stored on.
Exactly. That collision between the theoretical model and messy physical reality, that's where fortunes are lost and timelines just get destroyed. So today, we're dissecting Lesson 3, Execution Discipline, Process Analytical Technology, or PAT, and Robustness Across Scale.
Right, and we're asking the really expensive question here. Does your bioprocess succeed because your math is sophisticated, or does it succeed because you actually stress-tested the physics and, controlled the chaos?
I'll be taking the position that with rigorous execution discipline and modern control strategies, specifically PAT and quality by design, we can turn that chaos into a calculated risk. I really believe we can build an operating system for biology that guarantees quality.
And I'm taking the position that biology and, frankly, the laws of physics are the ultimate authorities, and they just do not care about your operating system. I'm here to argue that most of these models are what I'd call prediction fantasies. We ignore the messiness of inoculum history, mixing gradients, and then we act surprised when the scale-up fails.
Fantasy is a strong word, but let's dive into it. I want to start with this idea of QbD, quality by design, as an operating system, because the source material makes a really compelling point. A model isn't just some regulatory document you file away. It's the logic that should be running the plant.
Sure, but an operating system needs good inputs, right? If you type garbage into a command line, the computer crashes. My issue isn't with the concept of QbD, not at all. It's with the data we feed into it. We're building these, skyscrapers of mathematical modeling on a swamp of poor experimental design.
You're referring to what the material calls the ceiling of intelligence.
Exactly. It states it pretty clearly. Data quality sets the ceiling of intelligence for any model. And in early development, what are we all using? Shake flasks, microtiter plates.
Well, they're high throughput and cost-effective. You can't run 1,000 different designs in 5-liter benchtop reactors. You have to screen quickly.
No, you can't. But you also can't just pretend a shake flask is a bioreactor. They are fundamentally different universes. In a flask, you have surface aeration oxygen just diffusing in from the top. In a tank, you have sparging. You're bubbling it through the liquid. The hydrodynamics are completely different.
I'll grant you that. In a flask, the process is usually kinetically limited. The cells grew as fast as their internal biology allows because the environment is relatively gentle. In a tank, it becomes transport limited. The cells can only grow as fast as we can physically push oxygen to them.
Right. So if you build a model based on that flask data, you are modeling a biological reality that literally ceases to exist the moment you scale up. It's like you're training an athlete for a 100-meter sprint and then you throw them into a swimming pool. The muscles might be the same, but the physics of how they move are totally different.
That's a valid critique. But the solution isn't to just abandon early screening. The solution, and this is really where execution discipline comes in, is to use dimensionless groups to bridge that gap. We can't just copy parameters, we have to translate them.
Dimensionless groups. Okay, for everyone listening, let's unwrap that a bit. It sounds like academic jargon.
It's actually very practical. We look at ratios, for instance, the Damcollier number. It compares the speed of the biological reaction to the speed of mixing. Or we look at power input per unit volume. If we can match those key ratios between the flask and the tank, then we're not just guessing anymore. We're ensuring the transport regimes are comparable. We're translating the language of the flask into the language of the tank.
Theoretically, yes. But that assumes you know the reaction speed accurately. And this brings me to the hidden variability that just kills execution discipline every single time. The inoculum.
The starting material.
The cells themselves. We treat them like a chemical reagent. If I order sodium chloride from a supplier, it's 99.9% pure salt, every time. But cells? The source material makes a huge point about this. Inoculum age, viability, storage history. It all changes the metabolic state.
It's epigenetic memory. The cells remember where they came from.
Precisely. If your seed train, that whole series of flasks you used to grow up the cells before the main tank, if it's 12 hours older today than it was last week, those cells are different. They might be in a different growth phase. They might have switched their metabolic pathways. They might be, priming themselves for starvation.
And when you put them in the production tank, they behave differently. And the engineer immediately blames the pH sensor or the feed rate.
Right. You attribute the change to your design space, but actually you just started with tired cells. So you're modeling noise. You're trying to fit a curve to a ghost.
I agree this is a massive problem, but I see it as a failure of execution, not a failure of the model concept. This is why I argue for treating the inoculum as a critical material attribute, a CMA. We need strict specifications, defined passage numbers, specific growth phases at harvest.
Okay, but let's be honest. In how many labs is that actually done rigorously? Usually it's thaw the vial, wait 2 days, and hope for the best.
That's why we're having this debate. To shift that mindset, if we standardize the thought procedures and control the seed train timing, we turn that noise into a controlled input. We can't just throw our hands up and say, oh, biology is mysterious. We have to discipline the biology.
Discipline the biology. That is a very engineer thing to say. But let's look at another area where engineers try to force order on chaos. Missing data.
Ah, the failed runs.
The source material is scathing on this point. It describes a common bad habit. A run fails, maybe the agitator stopped or the pH drifted, and the scientist just says, oh, that was a mechanical failure. Exclude that data point.
Well, if the agitator stopped, the data is compromised. It doesn't reflect the biology we are trying to model. It reflects a broken motor. Why would I want that skewing my metabolic model?
Because you're assuming the failure is random, but often it's not. Equipment fails more often at the boundaries of the design space. Maybe the high viscosity at high biomass caused the motor to trip. If you throw that data away, you are erasing the evidence of fragility.
It's survivorship bias.
Exactly. You end up with a model that looks perfect because you deleted every single time it failed. You are lying to yourself about how safe that operating region really is.
Okay, I see your point. A failed run is actually the most informative data point because it shows you exactly where the cliff edge is. If the motor trips at high biomass, that is a constraint we need to model, not ignore.
Yes. A robust process isn't one that works only when everything goes right. It's one that survives when things go wrong.
Okay, I'll concede we need to be much more transparent about failed runs. But let's pivot to the solution I'm advocating for, the sensory system. The material talks about Process Analytical Technology, PAT.
Here we go. The shiny toys.
Not toys, eyes. Think about how we traditionally run these processes. We're flying blind. We take a sample, wait 24 hours for the HPLC results, and then realize we should have fed the glucose yesterday. PAT changes that. It gives us real-time visibility. Raman spectroscopy, near-infrared, off-gas analysis.
I love the promise of PSAT. I really do. But I am deeply skeptical of the implementation. Let's talk about soft sensors.
Soft sensors are brilliant. We use the data we can get easily, like spectral data or off-gas readings, to calculate the things we can't measure directly, like biomass or product titer. It lets us control the process based on outputs, not just inputs.
But how do you get from a Raman spectrum, which is just a squiggly line of laser light scattering, to 3.5 grams per liter of antibody?
You use math, chemometrics, principal component analysis?
Yes, we build a statistical model that correlates the spectrum to the concentration. And that is the trap. The material warrants explicitly about feature extraction and overfitting. You feed a high-dimensional dataset into an algorithm, and it will find a correlation. It might find that your product titer correlates perfectly with, I don't know, the background fluorescence of the plastic bag it's in.
That is a risk with black box models, sure. If you just let a machine learning algorithm run wild, it'll find nonsense patterns.
And then you run a new batch, the plastic vendor changes the bag slightly, the fluorescence changes, and your soft sensor tells you 50 grams per liter, when you actually have 0. You are flying the plane based on a hallucinating instrument.
That's why we need to focus on interpretable features, causal links. We shouldn't just look for patterns. We should look for the specific spectral peak that corresponds to the carbon-hydrogen bond in glucose. If we anchor the model in chemistry, not just statistics, it becomes robust.
Ideally, yes. But the temptation is always there to use the rich data because it gives you a higher R-squared value. It looks better on the PowerPoint for management. Look, our model predicts perfectly. It's hard to tell your boss, I'm using a simpler, uglier model because it's less likely to lie to us later.
True. But this brings us to the real payoff of PAT, control logic. I want to emphasize it, if we trust the sensor, we can move to feed-forward control.
Explain that for the listeners. Feedback versus feed-forward.
Okay, so feedback is reactive. Oh, the pH is low, add base. By the time you act, the cells have already felt the stress. Feed-forward is proactive. It's the cells are consuming oxygen at this rate, which means they are about to run out of glucose in 10 minutes, so I will increase the feed rate now.
So you're anticipating the need.
Exactly. This reduces system fragility. We aren't forcing the cells to conform to some fixed recipe. We are adapting the environment to meet the cell's needs in real time. Adaptive DO control, adaptive feeding. It's like cruise control for a bioreactor.
It sounds elegant, but I worry about the complexity cost. A smart system is incredibly fragile if a sensor fails. If your Raman probe drifts by 1% and your control logic dumps 50 kilos of glucose into the tank because it thought the cells were hungry, you've just ruined a $2 million batch.
So what is the alternative? Blind execution? A fixed recipe that guarantees variability because biology always varies?
Sometimes a fixed feed rate is safer because it can't be fooled by a bubble on a probe. But I agree with you. Adaptive control is the future. We just need to be realistic about sensor noise. We need fail-safes.
I think we can agree that the goal is consistency. But let's talk about what we're trying to achieve with that consistency. The material contrasts sharp peaks with robust plateaus.
Ah, this is my favorite part. The seductive peak.
We've all seen it. You run a design of experiments, a DOE, and you find this tiny, magical combination of pH, temperature, and feed that gives you this massive yield.
And the development team high fives. We hit 5 grams per liter. But that peak is a needle. If the pH drifts by 0.1 or the temperature shifts by half a degree, the performance just collapses.
Exactly. In a lab, you can hold that peak. You have extremely tight control. But in a manufacturing plant, forget it. Sensors drift, valves stick.
That's why I say development teams are often their own worst enemies. They hand over a process that is technically optimal, but operationally impossible. They hand the manufacturing team a grenade with the pin already pulled out.
So the goal of QbD execution has to be finding the robust plateau, a region where performance is high and stable, even if the yield is, say, 5% lower than the peak. If it's flat, if I can drift a little and still get the same result, that is the superior process.
And how do we prove we actually have a plateau?
Validation. We run confirmation runs, not just at the set point, but at the edges of the design space.
But simple confirmation isn't enough. We need sensitivity analysis. We need to know if the risk is symmetrical.
Go on.
We'll take feeding. Maybe underfeeding by 10% is fine, the cells just grow a little slower, but overfeeding by 10% might trigger the Crabtree effect.
The shift to overflow metabolism.
Yes, the cells stop breathing properly and they start dumping toxic byproducts like lactate or ethanol. The culture crashes. So the risk is asymmetric. One direction is a stumble, the other direction is a fall off a cliff.
That's a crucial insight. A simple plus-minus range on a spec sheet doesn't capture that cliff edge.
No. You need to know that the cliff is on the right and the meadow is on the left. So you set your target closer to the meadow. You bias the set point away from failure.
And that leads us to the biggest cliff of them all, the physics of scale, the dissenter's ultimate weapon.
The reality check. We have to talk about gradients.
In a 2-liter lab reactor, mixing is almost instantaneous.
Right. You add a drop of base, and zip, it's mixed. The whole tank is homogeneous. It is a single environment. But in a 20,000-liter tank, it's a swimming pool. It can take minutes to mix.
And that creates zones.
Zones of terror, I call them. Near the feed pipe, you have a huge sugar spike. That's osmotic stress. Far from the impeller, you have oxygen depletion. That's starvation.
So the cells aren't just sitting in a nice warm bath. They're circulating. They're traveling through these different zones.
It's a roller coaster. Every few minutes, a cell goes from feast to famine to acid shock and back again. And that triggers stress responses you never saw in the lab.
This is where the model usually breaks down.
This is where the model dies, because your model assumes concentration equals X. But in reality, concentration is X here, Y over there, and Z somewhere else. The average condition that you model doesn't actually exist anywhere in the tank.
So how do we fix this? We can't change the physics of large tanks.
We can't, but we can change how we model them. We have to embed scale-up physics early. We need to measure the KL, the volumetric mass transfer coefficient. We have to know the oxygen transfer capacity of the big tank before we even start.
And the material suggests using scaled-down simulators.
These are clever. You take your little lab reactor and you attach a plug flow loop to it. Basically a tube where the cells sit without oxygen for a minute before circulating back. You force the lab cells to ride that same roller coaster.
So that way you aren't guessing. You are breeding cells that are tough enough for the journey. You're validating the biology against the physics.
Exactly. But honestly, it's expensive. It's complex. So most people just rely on scenario testing, which, often just turns into prediction fantasy.
You keep using that phrase.
Because it's rampant. A prediction fantasy is when you assume your model holds everywhere. Oh, the model says if we lower agitation, the yield goes up.
But in the big tank, lower agitation means worse mixing, which means more gradients, which kills the batch.
Precisely. Real scenario testing means intentionally breaking things in the lab. What happens if the feed line clogs for an hour? What happens if the air compressor fails for 5 minutes?
You have to simulate the failure modes.
Yes. Validation shouldn't be a victory lap where you run 3 perfect batches and pop the champagne. Validation should be a stress test. You should be asking, how close are we to the edge? If you don't know where the edge is, you aren't safe.
I agree. And this aligns with a modern regulatory view of continuous verification. It's not a one-time stamp of approval. We keep monitoring. If we see the KLA decreasing over time because the sparger is getting fouled, we adjust.
And that requires humility. It requires admitting that the model isn't perfect and it might need to evolve.
I would say the model should evolve.
Look, let's touch on one final physical limit, oxygen transfer. This is usually the hard constraint, isn't it?
It is. In a lab, you can spin the impeller at 1,000 RPM. You can pump in pure oxygen. You can support massive cell densities.
But if you spin a 20,000-liter impeller that fast, the tip speed is so high you start shearing the cells apart. Or the power draw just blows the fuse on the whole building.
So the oxygen transfer capacity per unit volume drops as you scale up.
Which means your design space has a hard ceiling. If your model predicts that more biomass equals more product, but the big tank literally cannot breathe enough to support that biomass, you have a broken model.
I would argue you have an incomplete model. If you use QbD correctly, you incorporate the physical limits of the equipment into the design space itself. You don't say optimal is X, you say optimal is X given that the oxygen uptake rate cannot exceed Y.
Fair point. If you constrain the model with reality, it works. But that brings us right back to the human element, the operator.
Execution discipline.
You can have the best PAT, the best KLA calculations, the most robust plateau. But if the operator thaws the violin correctly,
Or if they don't calibrate the pH probe properly.
it's game over. The material emphasizes that consistent application of SOPs, standard operating procedures, is what determines whether theoretical reproducibility becomes actual reproducibility.
And this is why I see execution discipline as the tool that turns QbD from a theory into reality. It isn't just math, it's procedure, it's data standards, it's training. It is the discipline to do the boring things perfectly every single time.
And it's honesty.
Honesty?
Intellectual honesty. Admitting what we don't know. Admitting that the ceiling of intelligence is limited by our noisy data. Admitting that scale-up is going to introduce gradients we can't fully predict.
I think we can find common ground there. The goal isn't statistical perfection, it's operational stability.
Exactly. I don't want a sharp peak that exists only in a computer. I want a boring, stable process that survives the chaos of the manufacturing floor.
And to achieve that, we need both. The sophisticated eyes of Pat to see what's happening, and the gritty discipline of stress testing to ensure we can handle it. We have to respect the model, but verify it against reality.
We need to stop trusting the models blindly and start making them earn their keep against the hard laws of physics.
A fair challenge. There's always more to explore in how we simulate that reality. But for today, I think the lesson is clear. Respect the scale and discipline the data.
And keep questioning the model.
Thank you for joining us on The Debate.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.